Okay, so today what I'm gonna be doing is talking about some work that I did while I was a assistant professor at the University of Michigan in the biophysics department. And this is work that we haven't discussed before. And so here what we'll talk about is exploring structure sequence space using image-to-image translation. And so this is work that was done in collaboration with former graduate student, Sarah Witt. She actually recently defended, so she's now Dr. Sierra Witt. And so the plan is to talk about this work, and if we have time, talk a little bit about my trajectory, my career trajectory, because it's been from grad school to industry, back to academia, and back to industry. So it's kind of an interesting path. Some of you might find interesting. So let me jump right in. So when we think about RNA and RNA structure, right, there are multiple levels at which we can describe the RNA. So we can start by talking about the sequence of an RNA, which gives us a sense of how individual nucleotides are connected, right? So how the string of nucleotides that form a particular RNA. Now, because the nuclear bases are able to base pair, an RNA can fold back onto itself, and you can arrive some secondary structure which contains information about the sequence plus the base pair contacts between individual nucleotides. So the next level of structural comes at the 3D level, which describes how each of the nucleotides, each of the atoms in the nucleotides are arranged in three-dimensional space. So the most complete form of the information you can have is in the three-dimensional structure, because it contains information about the relative positions of the individual atoms in the RNA and the sequence. So a lot of the work that has been centered around RNA structure prediction is mostly attempting to go from sequence all the way to 3D structure, right? And there's great interest in understanding 3D structure naturally, because a lot of the functional capabilities of RNA is kind of rooted in their ability to adopt specific three-dimensional structures. And in addition, some RNAs are actually emerging as drug targets. So the company in which I work at now, Arrakis, is focused on identifying small molecules that can bind to RNAs, right? And so understanding the three-dimensional structure is really important. Today, what we will talk about is not 3D structure prediction, but the inverse problem, right? So this is this idea of, starting from the secondary structure, filling in the missing information and getting access to the sequence that would stabilize a particular structure. And so the design problem, as you guys are all aware of here, is the challenge of determining the sequence or a set of sequences that stabilize a target structure. Now, this kind of representation showing you, just the RNA structure on the left without any sequence information, and then on the right showing you the sequence, kind of is actually hinting you to the way we are thinking about this problem. It's almost like what we're trying to do is impute missing information, right? Start from low information and go towards higher information and filling in the missing details. So there are a number of frameworks in which you can think of the RNA design challenge. One is a thermodynamic framework, where the goal is to identify a sequence or sequences with a minimum free energy structure that matches the target. The other is one where we're using kinetics, and the goal here is to identify sequences with structures that fold to the target at a particular rate. So not just getting the right structure, but targeting a specific rate of folding. So the one we're going to talk about today is really about this thermodynamic framework, where what we're trying to do is really just simply identify a collection of sequences that allow us to adopt a particular target structure. So in our hands, the way we kind of thought about this problem is a two-step problem. The first is sequence generation, right? So that means that what we're trying to do is generate a set of sequences that are likely to allow the RNA to adopt the target structure. And then there's an additional refinement step, which you can think of as sequence scoring, right? So you have a bunch of sequences, you might want to prioritize some sequences over others if you had to make decisions as to which one you might actually want to synthesize and test in the lab. So today the focus is going to be on sequence generation. So what I'm going to talk about in the first part here is this idea of using machine learning model, right? And so what we want to figure out is whether or not we can teach, whether or not we can teach models to implicitly learn structure sequence relationships. And I say implicitly because we're not going to actually directly reference the RNA design problem, but we're going to actually try to map the RNA design problem onto something that is familiar to a machine learning model, and then essentially use that as a sequence generation tool. So just a quick bit about machine learning. I'm pretty sure most of you have heard of it, have thought about it before, but just to make sure we're all at the same level, we're just going to talk about machine learning for a bit. So essentially what you're trying to do with machine learning is to try to mimic what humans do, right? And so is to learn from historical data such that you increase your performance over time. And the kind of machine learning models that we are actually interested in here are these machine learning models that make use of images. So the idea is that you give the model some input and then transform that input to give some output. And here, the input is an image and it gives you another image. In this example here, it's actually adding in some missing detail. So this is what you would call an image-to-image translation problem. And so the approach that we're going to use for in the inverse design problem is really is motivated by these kinds of models that take an input image and then output an image. So another example is shown here where you actually add color to an image. And so you go from black and white to color, which really maps onto the problem that we're gonna talk about. And others allow you to take a sketch, an outline of the image and fill in the missing detail. So it's this idea that you can use machine learning models to operate on images and filling missing details. So how can we leverage for the challenge that we're interested in? So the first thing we attempted to do is actually encode RNA secondary structure as a 2D image, right? So the idea is that you take the secondary structure and you create the dot matrix that contains the contacts between individual nucleotides. And then you color each of those positions based on the identity of the nucleotides and or the base pairs. So in this image here, the diagonal elements contain unpaired nucleotides, and the color is given using this palette we have here. And those are the off diagonal correspond to base pairs, and the identity of base pairs is linked to a specific color. So what we've done here is take secondary structure and encode it into a 2D image that is colored. And so one could imagine then if you remove the color, all you have left is just the structure, right? So going from this black and white decolorized image to a colorized image really is going from just structure to sequence. And so you can imagine training machine learning models on pairs of decolorized images and colored images, and then using that model to actually do the inverse design, right? So what would happen is that you would take this target structure, feed it into your machine learning model, and you add the color back to this image. And from that image, you can extract the sequence because we have a specific palette that we use, right, to be able to link color to sequence. So what we've done here is really map the inverse design, RNA inverse design problem onto an image-to-image translation problem. And we've stripped away all of the physics from the problem, right? It's just purely an image-to-image translation problem. And what this allows us to do is to actually make use of machine learning frameworks that have been applied. For example, in this case, right, taking a black and white image and adding the color to an RNA inverse design problem. So I'm gonna skip a lot of the details about the machine learning model in terms of how it was trained. We essentially what we did is that we generated about 6,000 random sequences, folded them using RNA structure from David Matthews. And then based on the secondary structure we created, we turned each of them into a colored image and a decolorized image, right? And then we trained the model, and then we deployed the model. So what I'm going to talk about next is how we challenged this model, and we're going to compare its performance. And so essentially what we did is take about 174 RNAs for which we know their structure, and removed the sequence information, essentially generating these black and white images that just contain information about the secondary structure, added the color back in using our model, and then asked whether or not the RNA, the sequence that we extracted from those images allowed us to fold back to the target structure. And to quantify our success, we use this PPV, which just essentially allows us to assess the extent to which we can recover the base pairs in the target structure. So the PPV, as you know, higher values approaching 100 correspond to good predictive power, whereas values close to 0 means poor predictive power. And so what we wanted to do is actually compare it to when we just use random sequences. And you can see in this image on the right that our PPVs using our tool, which we call RNA Explorer, or tentatively, it's kind of shifted towards higher PPV values, right? So it suggests that training this very simple model, right, that's based on rendering RNA structure, secondary structure as images, allowed us to actually design RNAs that outperform just random design. And this is, probably the most basic of the assessments we can do, but it was an important step here. I should note that it's interesting that we trained the model using synthetic data, right? So just random sequences, right? And then we actually tested them here using actual RNAs that we know are secondary structures. And so we were actually quite pleased to see that we were getting some enrichment in higher PPV structures. So I'm going to go a little deeper for a few of these RNAs. And so one RNA here is the fluoride riboswitch. So the riboswitches of these RNAs are able to recognize ligands and lock the riboswitch into a particular conformation. And in so doing, regulate either transcription or translation. And so this is an example of the riboswitch that's regulated by the fluoride ion. And then you have the SAM riboswitch, which is another RNA that's able to recognize SAM. And it too can turn on and off gene expression by binding to the SAM, which is this cognate ligand. So what we did is to actually generate sequence designs that are consistent with the secondary structure in these two test RNAs. And so what we did is then created web logo to kind of capture the sequence variation at each position in our designs. And so what you're seeing here on the top would be the distribution at each position for our designs, right? So I think we had about maybe 2,000 designs go into this web logo that you see at the top. And the next line, this line here contains, or this row here contains an example of the sequence from the crystal structure. And then on the bottom, we have the consensus sequence. And what you can see, the positions with the asterisks correspond to cases where there was very good correspondence between the sequence distribution at a particular site and the consensus sequence. And so you can see that it's not perfect, but there are quite a number of sites that were able to recover the correct sequence distribution. And the same is true for the SAM riboswitch as well, right? Again, here we generate thousands of sequences, so this web logo corresponds to the distribution across thousands of sequences. Now, this kind of leads us into one of the challenges we immediately ran into, right? Which is that, I showed that indeed, compared to random, right, our designs tend to have higher PPVs when you try to pull them into the target, but they're not perfect, right? So we actually run into a selection problem where we can generate many designs, as many as we want, right? But then we have this problem choosing the right one, right? And so that led us into thinking about ways we can actually do that second step, which is to score individual sequences. So what I'll talk next is about one approach one can take that really relies on atomic simulations. So going a step above just looking at secondary structure, now you're considering the three-dimensional structure. And so you could use molecular dynamics to do this, right? And so molecular dynamics is essentially a physics-based 3D modeling approach where essentially you use Newton's equations of motion to be able to describe the motion of each atom or each particle in your system. And so these molecular dynamics could operate on an all-atom level where you try to model each atom explicitly, or you can do more coarse-grained representations where you use a simplified representation of your monomers. So the coarse grain method that we use is called SimRNA. It's a tool that was described in this publication. So it's not something that we developed, but it's a tool that is widely used. And so it's used as a close grading tool for folding RNA, and it utilizes a Monte Carlo-based confirmational sampling technique. And so what we did is actually attempt to use these simulations and analyze the resulting data to try to see if we can use it to score individual sequences. So the first thing we did is to actually just confirm that in our hands, we can actually fold RNAs. And so we were focused mostly on pseudoknotted RNAs. So these are RNAs where we know there are pseudoknotted interactions. And for the most part, the PPVs we get from the folded structures are pretty high, with the exception of this set here, which corresponds to cases where we had about 4 pseudoknotted interactions. Don't really have a good explanation as to what's special about 4. I don't think anything is special. It might be just a sampling issue. But for the most part, we can use SimRNA to fold most of these 40 pseudoknotted RNAs. So now that we feel confident that we can fold these RNAs into their target structures, we want to actually take a look at the sensitivity to changes in sequence. So the first kind of changes we applied were pretty dramatic changes where we take the sequence and we scramble the sequence, right? Or we take the sequence and we just generated random sequences. And so when we do that, if we use random sequences, right, we look at a PBV, when we actually target a known structure, we get pretty low PBVs. And then we use scrambled sequences, we also get low PBVs as well. So by comparison, if you take the wild type sequence and you target the corresponding secondary structures, the PBVs are about 75%, the mean across the 40. And so this gives us a sense that, okay, if we make modifications to the sequences, pretty dramatic modifications to the sequences, and then we try to fold them to the target structure, then we can actually keep track of the PBVs that you get and use that as a way to score individual sequences. Now, one of the things that we noticed is that once we try to do more subtle changes, then the PPVs no longer were useful for this type of analysis, right? So imagine we take the original sequence and we just change the identity at one position. We're having some issues using the PPVs. And so what we did is a slightly different analysis. So one of the things you can do is that, once you provide a target structure to SimRNA, it can also tell you what's the restraint energies. And so the restraint energies capture the extent to which you need to add additional energy into your system to allow you to fold into a target structure. So the higher the restraint energies, the less compatible the sequence is to your target structure. And so what you can imagine if you had a set of 3 sets of sequences for a particular target, those that are most compatible should show lower folding restraint energies, whereas those that are least compatible should show high restraint energies. And so if you were to draw a line at any folding step, you can actually make a plot like this, which kind of shows you the restraint energy as a function of folding time. And cases where you have lower restraint energies correspond to cases with most compatible with your target. And so you can imagine doing this kind of analysis to assess the quality of individual sequences. And so that's what we attempted to do. And so I'm just going to walk you through an example here. So this is a restraint analysis that we did when we were looking at the fluoride ribose switch. And we could look at the specific sites in the RNA. So for example, at position 45, right, this position here, it's base paired to this A here. And you can see that when that position is U, right, that's a wild type sequence, that has the lowest restraint energy, which makes sense because it's base paired to an A, sorry, right? And if you look at the residue, which is base paired, right, that position is where you have A37, right, gives you the lowest restraint energy as well. And once you make mutations, let's say you go to a U at that particular site, you create a UU mismatch and you see the restraint energy is much higher. And so this kind of analysis suggests that you can actually make introduce these small mutations and then assess the restraint energies as a proxy for the compatibility of your mutant sequence to that target structure. So these are quite obvious. These are cases where you have base pairs, right? And then we know the base pairing rules. So this doesn't really add a lot of value, right? But there are instances where you do see some interesting trends. And so these sites that are shown here now correspond to sites that are not base paired. And so in this case here on the upper left and the lower, sorry, the upper right and lower right, you see cases where based on a restraint analysis, you can't really make any real, there seems to be no real preference, right, for U, G, C, or A. An interesting one is this position 6, right? Where you do see some preference, right? So it seems as if there's a gap between the restraint energy profiles that we get for U's and C's versus A's and G's, right? And so the question we had is one of, okay, why is there a preference for U or C at this position, right? So this kind of speaks to the need to actually now think about three-dimensional structure, right? So what I'll show you here is our attempts to rationalize what might be happening. And so I'm showing you the surface of the RNA where at position 6, I removed that nucleotide, right? And then I'm going to put it back in. And you can see that it kind of fits neatly into this position here, right? Now, once you make mutations, you can model in mutations to, let's say, a C or an A or a G. You actually start seeing clashes showing up, and the clashes are most pronounced for an A and G, right? And this probably explains why we see this kind of profile where, you know, there is a difference in the restraint properties when you have a A or a G versus a U or a C. Now, one thing that we know about this particular site is that functionally, this fluoride ribose switch, if you make a change, if you change this U to C, it has 100-fold impact on the function of the ribose switch, right? And that's not captured in our analysis here, right? And so this actually speaks to the need to actually think about not just secondary structure, not just 3D structure, but also considering the impact of external factors. And I didn't put it in here, but there's actually a magnesium that sits right in this position here, right? And it interacts with this, this uracil. And converting that to C actually disrupts, it's believed to disrupt a key hydrogen bond, right? And that destabilizes the RNA, right? So without considering that external factor, it would be really, really hard to rationalize this sequence function relationship at this particular site. So I think this is a nice way to kind of bring home the point that, you know, in addition to thinking about secondary structure and just the structure of the RNA itself, we also need to really consider external factors like magnesium binding. And so with that, I'd like to conclude. And so what I've described is very, very initial results, right? And it indicates that the RNA inverse challenge can be cast as an image to translation problems. This is a typo. Our image-based sampling of sequence condition on target structure yield designs that agree with some of the available phylogenetic data. We then show that you can use post-grain simulations as to be able to explore sequence structure relationships. And exploring sequence structure relationships will ultimately require all-atom resolution and consideration of the role of stabilizing ions, right? And so this is more of a comment rather than something that we clearly demonstrated. So future directions that one could imagine taking with this work would be one, retraining the model using larger data sets of experimentally informed sequence structure pairs, utilizing a chemistry-aware color palette. Right now, we didn't really try to encode any relationship between nucleotides and their colors, such that, you know, purines should have colors that are close to each other versus pyrimidines. We didn't do any optimization of our palette. That's something one can imagine doing. And then implementing a complete image then simulation-based pipeline to explore sequence structure relationships. So that means using this image-based sampling to generate an initial pool of designs and then filtering those out using something like SIMRNA, as we showed. That would be an interesting pipeline to implement and test. And then one thing that's missing right now is that the tool, we haven't made the tool accessible to the public, right? And this would be really important because, you know, if I have time, I'll chat about it. Let's see how much time I have. Actually, I don't have much time though. now that I've exited academia, some of these tools that we've developed would require others to actually take them and run with them. And so we would definitely want to make this public so that others can take a look at it. So I don't have time to do the other bit I was gonna attempt to do. So what I'll do is just take questions at this stage.
Hey, Aaron, thank you. We have a surprise guest with us. Sierra is here.
Yeah, I didn't actually tell you I was coming.
There you go.
Yeah, that's me.
That's her. Okay.
Jonathan, did you have a question?
Yeah, I'll ask a quick technical question based off of my memory of the machine learning class I took. I could imagine a difficulty with the method that you used would be encoding when you have different structural motifs kind of in different locations on your matrix, is actually getting the relationship between those kind of motifs encoded in addition to the motifs themselves, which, there might be a particular motif that is actually kind of entirely kind of spread throughout the image. Are you able to manage that well enough with some sort of convolution techniques or using well-designed kernels or something like that?
Yeah, so the underlying model is a convolutional neural network that is able to, look at sub images and get a sense of the features in those sub images. And so that is handled using the convolutional network. The other way it would be to actually translate your images, right, in that space so that the motifs show up at different locations in your image. But a convolutional neural network architecture is able to handle that.
And one other thought that I had was I would be interested to see this run against. We have a structured or a sequence design benchmark that we have published as part of Eterna called the Eterna 100, and I would be curious to see how well this would perform. It might need to be retrained against Vienna RNA fold, but I would be curious just to see how it would stack up in that benchmark.
Yeah, I think once it's retrained, that would be a fair comparison. Yeah.
So I'm curious, you're saying about, I know you're now at Arrakis, and you mentioned continuing the work. Sarah, are you continuing the work, or someone at Michigan continuing the work?
Great question. I don't know. As Aaron mentioned, I defended like 2 weeks ago. And now that Aaron has left the university, I think we have the goal of, as he mentioned, publishing a lot of this making this tool available. And I think we're not sure exactly what comes next for this project, but we're still really excited about it. So hopefully, but you never know, I guess is the answer.
Yeah, sure. I was just curious if you knew anything more. And would you see potentially having a large data set of thousands of verified pseudonaut structures being useful?
Yes.
Okay, because that's going to be one of our next projects potentially. So get back to us, Ever, if you want to know more about that.
Cool.
Thank you so much for the talk. That was, I certainly found it fascinating.
Thank you.